Okay, I know we've gone through this ad nauseum, but we're just going to quickly discuss it. So now in week 3, we've had quite a look actually at we've done introduction to cell biology. We've had quite a look at some cell structures. We've kind of moved through that, and we're now moving into cell function. Now I've kept it green because we're going to look at cell membrane and the actual structure of the cell membrane to start. So I've kept it green, but really we're starting to move into cell function and we're moving towards respiration. And you can tell because the last of our lectures is last of the sub-lectures is enzymes, and enzymes are really important for respiration and photosynthesis. And so that's kind of the back end of this week before we then transition into respiration and we start talking about the chemistry of respiration. Okay, little smidgen of revision because this is a repeat slide from the previous week, but I just want to go over it because this is important to what we're transitioning into. So we have these things called plasma membranes, and they are the skin of the cell. So they separate the inside of the cell from the outside environment, and that's important because we need to, or our cells need to have different densities of molecules inside the cell to the densities of molecules outside the cell, because the internal environment may have different requirements, different chemistry, because we need to have slightly different reactions occurring, and we might have to catalyze and speed up those reactions. So having that cell membrane is really important. As a consequence, the cell membrane actually controls the movements of things into and out of the cell, and it is what's referred to as semi-permeable. So it allows certain things in naturally through diffusion, and we're going to talk about that transport later. But other big particles it might not just allow in, and it might require it to actively move them inside because it might need to have a higher density inside than outside, and so it needs to control that movement. And now, so we've talked about a little bit about that, and we're going to move into these what's referred to as the distinct structures that are specific to different parts of the cell membrane, and that's what we're working towards. Now remember that cell membranes are common to bacteria, so the prokaryotes and the eukaryotes, and it's one of those, just those 3 things that are consistent to both prokaryotes and eukaryotes and all cells is they have these cell membranes. And you can see the animal cell up the top here, this has a cell membrane, and this is a plant cell, has a cell membrane. Now it's important to remember we're not talking about a cell wall. This plant cell also has a cell wall. Cell walls are not made of the same things as cell membranes. They are far more solid. They're not as fluid as a cell membrane, which we're going to talk about. And that's the reason that plants, when they die, stay standing. Whereas because you and I, although we're eukaryote like plants, we don't have cell walls. So the consequence is when we die, we turn to mush because our cell membranes start to fall apart and are digested by other organisms. Okay, so just quickly again, this is another slide you've seen before, but I just wanted to remind us and do a smidgen of revision. All this is saying is that we're talking about the cell membrane, so the skin for the whole cell. However, inside the eukaryotic cells, we also have these membrane-bound organelles. And although when we're referring to the membranes now, we're going to be talking about cell membranes, and they have some slightly different particles that won't be present on the membranes of the membrane-bound organelles in every situation. The general structure is relatively consistent. So when we're talking about membranes and we're talking about membranes in those organelles, we are talking about that phospholipid bilayer, which is consistent with the cell membrane. So mitochondria, for instance, they have 2 phospholipid bilayers. They have 2 membranes around them. Chloroplasts have two membranes. The nucleus has 2 phospholipid bilayers. Vacuoles have a membrane. Rough endoplasmic reticulum, smooth endoplasmic reticulum, and Golgi apparatus pretty much are a membrane. That's what they are. And vesicles are just kind of a membrane really around a space. So just remember that we're talking about membranes for those as well. Okay, so this is what I want to do. I'll give you some diagrams and we'll discuss the features of the diagrams. And then what we'll do is I'll break it down into the specific structures and we'll just have some notes from those specific structures. So I want to discuss the structures in a way that gives you a little bit of a breakdown of what they do in association with the diagram. And then I'll kind of be repeating when we go back to the slides specific to those structures. But I think it's really important for different types of learners that's just visually seeing what they kind of look like. Remember that this is a diagram. It's not a picture. And then we can kind of break it down into the notes for each specific structure. But I want you to see them in association with the other structures in the diagram as well. Okay, so the first thing I want you to know is the present model at this time is what's called the fluid mosaic model for the membrane. Yeah, so fluid mosaic model. Remember that there are, and historically there have been other models for what a membrane looked like. And although we're not going to get into the history of the science, the development of the fluid mosaic model, you are certainly welcome to do so. A lot of textbooks have that information in them. It's relatively common that you would go through the history just to talk about the process of science. And it is a beautiful story of the development of hypotheses around what the membrane looked like, what the particles were, and then the disproving of that and some other evidence, and then the change to new models, etc. And it is really interesting. We don't have time to get into it within this course, but if you're interested, you're welcome to look into it. And it is, it's good science. It's very interesting science. The accepted model at this time, however, is what's called the fluid mosaic model. Now, we'll break that down. So fluid in this case refers to the fact that these phospholipids, the blue things, just see the little blue bump with the little tails there, the phospholipids, their association is quite fluid, so they're quite loose. The consequence is they actually move around, so the cell membrane can move, and the position of the particles, even the integrated proteins, like not this one, but the integrated protein over there, can actually move within the cell membrane, and they consistently move. And they don't just move in terms of their position in association with each other, but the cell membrane is generally relatively fluid. So the consequence is the cell membrane can move and compress. And that's why we can have things like amoebas, which are changing shape continuously. And look at a beautiful video, if you can, on YouTube of an amoeba, because it's an interesting single cell organism that's just continuously changing. And it's such a fantastic example of that loose membrane structure. Okay, so it's fluid. The reason that we use the word mosaic is because if you look at our membrane just over here, it's not consistent. It's sure, the phospholipid bilayer is there, the phospholipids are there, there's a phospholipid just here and a phospholipid here. And remember that this is facing inwards into the inside of the cell, and that's facing outwards to the outside of the cell. But you can see all these fantastic integrated structures. So this is a protein just there. You see the little yellow dots in there, the cholesterol, and you can see these integrated proteins, this one over here, the integrated proteins, and over on the other side there's a peripheral protein, and then there's a phospholipid as well. So there's all these fantastic parts kind of integrated into this membrane, and that's why it's called mosaic. So there's a whole ton of these integrated parts in the membrane, which is where the term mosaic comes from. Okay, really quickly. Phospholipid bilayer comes from this. This here is a phospholipid. So you can see the little blue, the big blue round head here, and then the two little tails just there. Now that is an expansion of these over here, which is relatively obvious, but the reason that it's called a phospholipid is this is like the phosphate head at the top of it, and this is the little lipid tails. So you have a phosphate head and the lipid tails, and the reason it's called a bilayer is because you can see there's the phosphate head and the lipid tail, and then there's the phosphate head and there's the lipid tail. So the lipid tails face each other, and then the phosphate heads face outwards, outside the cell, or inwards into the cell, but not each other, if that makes sense. And the reason for that is because you have what's called a hydrophilic head, or a hydrophilic end, and a hydrophobic end. The hydrophilic end, hydro, water, philic, means attracting or associated. The hydrophilic end is associated with water, and the hydrophobic end, hydro, water, phobic, fear of, or in this case, repelling. So the hydrophobic end is repelling water and consequently pushes away from the majority water outside and the more water inside. So they actually face each other. And then the hydrophilic heads, which is water associating, face outwards into the surrounding environment or inwards into the internal environment. So you end up with this relatively consistent membrane that is quite consistent and stays together because of this really interesting structure. And we're going to get into that a little bit further in future slides. Then the double layer of the lipids is quite fluid, so we talked about that, and it has all these proteins floating within it. It also has what's referred to as glycoproteins, which we'll talk about, glycolipids and cholesterol, and they're all integrated into the cell membrane. Okay, so to explain some of those parts we were just looking at, we have, actually I'll just take those back. So the first one, that one over there is referred to as a glycoprotein. So this is one of the ones I would like you to know. So we now have the phospholipid. You're going to need to be able to identify a phospholipid, and you need to be talking about the fact that it has this hydrophilic head and a hydrophobic tails. Now I need you to be able to identify the fact that you have these things called glycoproteins. Now you can see that glycoprotein over there looks a little bit like a tree. It extrudes into the surrounding environment. And the reason it's called glyco is it has a carbohydrate structure, and that's that kind of tree-like thing, a carbohydrate structure that communicates with things outside the cell. And then it has the protein that's kind of semi-integrated into the cell membrane. And it has an important role in recognition and immune response. So all I really need you to know is this, is that the glycoproteins have these kind of tree-like structures that are carbohydrates, and that tree-like structure allows the glycoprotein to communicate with outside cells, and it's used in recognition. So an external cell, you've cut yourself, and external cells like bacteria have invaded your circulatory system. What your body needs to do is identify that it's bad and that it's an invader, as opposed to your cells. Now, your cells can't communicate through discussion. They can't say, oh, well, I'm part of the body. And they can't see because they don't have eyes and it doesn't function that way. So what they are reliant on is these glycoproteins. These glycoproteins identify what is your body and what is not your body. So if your cells, if a white blood cell, a protection cell, runs into another cell, it can identify whether that cell is your body by identifying using these glycoproteins. So the role of a glycoprotein is for what's called cell recognition. So what I want you to know is that we have glycoproteins, they stick outside the cell, they have a carbohydrate tree-like structure at the top of them, and that they're to do with cell recognition. Okay. You then have these things called glycolipids. Now the glyco part is the carbohydrate top on it. It looks a little bit like a small version of the glycoprotein. And where the glycoprotein, the base of it, you can see that kind of protein at the bottom there. The glycolipid has a little lipid tail at the bottom here. It's a little bit harder to see. And these are associated with surface receptors, so they can receive particles from the outside, and they're also to do with membrane stabilization. So all I need you to know is that glycolipids have these little carbohydrate heads of them, and that they're to do with interacting with the outside world, and they might receive information from the outside world, and that they're to do with cell membrane stabilization. That's all I need you to know. Okay. Now, there are other proteins other than the glycoproteins. There's a whole bunch of them. In fact, they're one of the really common particles in the cell membrane. Now, I don't actually need you to know all of them, but there's a couple of them that are really important. And this is an example of them. This is what's called an integral protein. Now, all that means is it's integrated into the phospholipid bilayer. So this is the phospholipid bilayer, the blue round things with their little tails here. And you can see this protein is like a tube that runs through the phospholipid bilayer. So that's an integral protein because it is integrated through the phospholipid bilayer, and it works like a tube. And they can control the movement of particles, which we'll get into deeper in the next sublecture. But they can control the movement of particles, larger particles, into and out of the cell. So the cell membrane, in terms of the phospholipids, will allow things like water, small particles that aren't ions, so don't have a strong charge. They will allow small particles like water into and out of the cell membrane naturally, but some larger particles or charged particles can't do that naturally, and so as a consequence we have these proteins that integrate through the membrane and act like channels. So this one is an integrated protein, and that's what I would like you to get out of this, is that we have these proteins that work as integrated proteins. The other protein that's over that side over there that's sitting on the outside, that is referred to as a peripheral protein. Now, all I need you to know is that there are some peripheral proteins. That'll do the job. Peripheral just means edge. They're on the outside. They're on the edge of the membrane. Okay, a couple more things to say. The glycolipids also help to organize cells, but I don't need you to know that. That's just a little bit of extra information. And organize cells into tissues, so they just allow them to kind of orientate themselves. Now, this is what I was just saying before. The phospholipid bilayer allows some things like water to just move directly, to diffuse directly through the cell membrane. It doesn't need to kind of be helped through the cell membrane or have a tube. It'll just go straight through the cell membrane, and it'll move from an area of high concentration to low concentration. Yeah? So if there's lots of water on the inside of the cell and not a lot on the outside of the cell, then what will naturally happen is the water will just diffuse straight through the cell membrane to the outside of the cell. Now we're going to talk about words like diffuse, and we're going to discuss that later. But just know that water doesn't need to be moving through protein channels. It can just move straight across the cell membrane. Okay, so that's just talking about what we discussed about the fact that some of the larger structures like ions and carbohydrates, remember that ions are charged particles, and carbohydrates can move through these tubular proteins, the channel proteins that are referred to as integral proteins. Now, last but not least, is this beauty right over the other side there called cholesterol. And these we find in the membranes. Cholesterol, you see it has these kind of yellow dots. You can see these kind of cholesterol yellow dots there. And you can see at the back end of that far, cholesterol has a little tail off the back. You find cholesterols in the membrane, and what they do is they disturb the closely packed phospholipids, and they keep the membrane fluid, and that's really important because the fluidity of the membrane is really important, and the phospholipids can start to kind of solidify a little bit, and you need those cholesterols to make sure that it doesn't get too solid, that it still has that fluidity so it can interact with their surrounding environment more effectively, and it can respond to pressure changes, etc. Okay, so let's get into some descriptors. We've looked at the diagrams. Now we're going to quickly bust through the specific molecules again. We've kind of done this, but I just want to have it, I kind of just want to have it up there so you've got it a bit more listed. Okay, so a quick closer look at phospholipids. So we talked about this again, but I'm just going to repeat it. This is a diagram, well, it's a chemical structure for the phospholipid. These are the two fatty acid chains, the two tails on the back end of the phospholipid. So these guys are hydrophobic. So they're the ones that face inwards in the cell membrane. So they face like this. They're facing inwards in the cell membrane. And then you can see the phosphate head. So they are hydrophilic. The phosphate head, faces outwards to the surrounding environment or into the cell. And that's just the chemical structure of that blue diagram that you've seen before. the common diagram that you've seen before. Now, what's important about that is that these phospholipids are the most common particles, and that they have polarity for the hydrophilic head. It just means that it has a variation in charge across the particle, and non-polar for the hydrophobic tail, and it just means it doesn't have a lot of change in the charge across the particle. Okay, so we know what the particle looks like. Now we're just going to have a quick relook at what the structure is that responds to that particle. So you can see this is that hydrophilic head and the hydrophobic tails, and you can see that because those tails are hydrophobic, they face away from the water on the outside, and because there's also quite a bit of water in the cytoplasm, they face away from the water on the inside, with the consequence that the two hydrophobic sets of tails face each other. And then the hydrophilic heads, the polar heads, the phosphate heads, face either into the cytoplasm or out to the surrounding environment. And again, the reason it's called a bilayer is because you have these guys and these guys. So there's two layers, and this is naturally forming. In fact, if you drop oil in water, it'll associate into a ball as well. It'll turn into this little round ball. Drop some water when you're making spaghetti. You put a little bit of oil into the water, and you'll see it automatically goes into these ball shapes. Well, your cell membranes kind of do the same thing. Okay, so really quickly, that's just a repeat of that information, just a bit of a descriptor of how the hydrophobic and hydrophilic tails function. And now let's talk about proteins. So there's two types of proteins. There's the glycoproteins, and then there's the general membrane proteins. And we're going to start with the membrane proteins because they're so important and they're so interesting. Okay, so there's a bunch of different embedded proteins in the bilayer. There's a whole ton of embedded proteins in the bilayer, and they have diverse structures, and they have a whole ton of functions. All I need you to know, because it's really easy to get lost in all these different proteins, is that there are integral proteins and peripheral proteins. Integral means they go all the way through, and peripheral means they just sit on the outside. And it's the integral proteins that are so important to us. And all I really need you to know, because we're going to look at this a little bit further, is that they have a function in what's referred to as channels for passive transport. This one's a channel, like a tube for passive transport. So that one's a channel. You can see the tube goes all the way through. And that's just to allow those kind of larger particles or charged particles to move through. Or they can act as what's called a pump for active transport. So passive transport, i.e. it just naturally moves in from a place of high density to low density, or active transport, i.e. it's brought in by the cell through the tube. So just know that there are integral proteins, i.e. integrated into the cell membrane, and that's what's shown here. Here's the outside, that's the inside. They're integrated into the cell membrane, they go all the way through, and that those proteins can work as channels or pumps. And that's, look, there's a lot more involved there that we could get into, but that's all I really need you to know. Okay, the next group of proteins is those glycoproteins. We talked about it quite well before, but I'll just quickly repeat it. Glycoproteins, they have an attached carbohydrate chain. That's a kind of tree-like thing on the outside, and they play an important role in recognition. So your cell's ability to recognize other cells that are part of it, or cells that are not part of it. And that has to do with your immunity, your ability to defend yourself. And they act as receptors for hormones and neurotransmitters. So they can actually act as to react to hormones that hit the cell membrane that might be communicating things, or neurotransmitters for neurons. Just another note there is just remember that these membranes are not static molecules, they're actually fluid. So that means even these proteins can move around in position in the membrane. They can actually move in position in the membrane. Okay, the next one is cholesterol in the membrane. So cholesterol is a type of lipid, a fat, belonging to a group of substances called steroids. Cholesterol has hydrophobic and hydrophilic regions and is therefore positioned between phospholipids in the membrane. So if you go back to the diagram that I showed you before, you can see it has a little tail. That is the hydrophobic tail, and then the region, the head of it, is the hydrophilic head. So you can see here is the hydrophobic is this bit, so repels water. So this faces into the membrane, and the hydrophilic is this little red bit down here. This faces outside the membrane with the phosphate heads of the phospholipid bilayer. And so these integrate into the cell membrane. Now they don't work all the way through, they're just on one side, but there's on both sides of them, there's these molecules could be on both sides of the membrane. Now what they do is they disturb, so they break up the close packed hydrocarbon tails of the phospholipids, and they prevent them from acting as solids. So rather than that membrane becoming too stiff and too solid, the cholesterols actually allow them to be a little bit more liquid. So it allows them to be more fluid, and that allows them to be more stable. It actually allows them to stay stable rather than having solidity, which potentially means that they might break up under pressure. So it also reduces the permeability, the ability to move of hydrophilic particles such as hydrogen ions, for instance, sodium and hydrogen ions. So particles that are hydrophilic might struggle a little bit more to just move straight through the membrane. And you can see just the last little bit there, they're involved in this thing called endocytosis, which we'll talk about later. Now, what I need you to know about the cholesterol is just that they allow for the fluidity of the structure of the phospholipids. So if you can just remember that cholesterol has an effect on the fluidity of the phospholipids, that's about it. Cool? Okay, so membranes must be fluid to work properly, as where the membrane solidifies, its permeability changes the ability for things to move across it. The fluid nature of the membrane allows it to change shape in a way that would be impossible if it were solid, and the fluidity also allows vesicles to be pinched off, and we talked about vesicles previously, but we're going to talk about them in endocytosis in the coming sub-lectures, and membranes to then fuse with them and connect with the membrane. And that, ladies and gentlemen, is that. So very well done on sitting through that. These are some little bits of extra research that you could be doing as we've discussed in the face-to-faces and previously. I encourage you to have a look at some of these. There's some fantastic ones there, and they present things slightly differently. They might have some slightly different diagrams that might just connect for you. They might not. I know that some people have the complaint that some of these are too fast, and I get that. But look, you can just pause them like you can pause me. And I'd have a look at some of those, and I think there are some great ones there.